Boa noite a todos. Bem-vindos a este webinar internacional da ProDiet Medical Nutrition. Hoje recebemos um convidado muito especial, o professor Stephen Cunnane. Ele irá falar sobre implicações terapêuticas com o uso de corpos cetônicos nas doenças neurodegenerativas do envelhecimento. O professor Stephen Cunnane é PhD, professor titular do Departamento de Medicina da Universidade de Sherbrooke, no Canadá, e pesquisador no Centro de Pesquisa em Envelhecimento. Ele e seu grupo estudam se intervenções baseadas em cetonas podem desacelerar o declínio cognitivo associado ao envelhecimento e na doença de Alzheimer. O seu grupo foi o primeiro a conduzir estudos utilizando PET com duplo marcador para captação de glicose e de corpos cetônicos. E também o primeiro a demonstrar que o estado de energia no cérebro de pacientes com doença de Alzheimer pode ser melhorado pelas cetonas. O Dr. Cunnane já publicou mais de 300 artigos e 5 livros, incluindo 2 sobre o papel fundamental das cetonas no desenvolvimento do cérebro humano. O seu conceito sobre o resgate de energia cerebral pelas cetonas, em específico para tratar as doenças neurodegenerativas, foi objeto da sua recente revisão publicada na Nature Reviews Drug Discovery. O Dr. Cunnane foi eleito pela Academia Nacional Francesa de Medicina em 2009. Em 2016 foi homenageado pela Sociedade Internacional do Estudo de Lipídios e Ácidos Graxos e recebeu uma medalha da Sociedade Francesa também por suas pesquisas em lipídios, nutrição e saúde. Antes de chamar o nosso convidado, para aqueles que utilizarão a tradução simultânea, o procedimento é bem simples. Vocês encontram um botão com o ícone de um globo abaixo da tela de vocês e vocês podem clicar na opção português. A partir desse momento, vocês receberão a tradução simultânea para o português desta apresentação. So, Dr. Cunnane, thank you very much for being here with us this evening. It's a huge pleasure for ProDiet. I'll be back for the Q&A session, and now the stage is yours.
Thank you very much. Let's see how to... It is my pleasure to give this presentation this evening on behalf of ProDiet, and I will be talking to you this evening. Let's just get the pointer correct. About brain energy rescue with ketones and the implication for neurodegenerative disorders of aging. The emphasis will be on Alzheimer's disease. I work at the Research Center on Aging, and I'm a professor in the Department of Medicine at the Université de Sherbrooke in Sherbrooke, Quebec, Canada. This is in the eastern part of the country, just north of Brazil, north of Boston, directly in the USA, a small town of about 150,000 people. My disclosures for financial support of our work as both public and nonprofit from the Alzheimer's Society of Canada, the Alzheimer's Association USA, and in Canada, and from the private sector as well. We will be talking about issues of health today, about Alzheimer's disease, about an emerging therapeutic strategy. I am not a physician. I'm not a dietitian, and this does not constitute advice to anyone for any medical condition. So as an overview, these are the topics that I will be discussing with you this evening. We will be talking principally about Alzheimer's disease, which we abbreviate AD, and with specific emphasis on the early stage of Alzheimer's disease, which is called mild cognitive impairment or MCI. I'll be discussing brain fuels, which means glucose, but also ketones. And I will discuss them in the context as I refer to the model of the hybrid car running on electricity and gasoline. And this is like the brain, which is running on two different fuels as well. These molecules are important as fuels, but they're also important for the lipid structure of the brain and for the wiring, for the connections, as you will see from my presentation. Perhaps the most important point is that there's a physiological basis for studying how ketones might be beneficial in the aging brain based on their importance for infant brain development. So I will take some time at the beginning of my talk to review this subject, and then we will discuss the clinical trial results. So let's start with the infant, because this is where the importance of ketones was first understood in the 1950s. The reason ketones are important for infant brain development is because the infant brain has a very high energy requirement. So its body weight about 3.5 kilograms at birth. The brain weighs about 400 grams, but it is consuming 74% of the whole body energy requirements of the infant, which is a huge amount. It's 3/4 of the energy required by the baby at birth is going to its brain. And it's not even working very well, the brain at that stage. Compared to adults whose brain is about 3 times bigger than in a baby, but the consumption of energy is about 23%. So the proportion goes down. So the problem for the infant is that it has a high energy requirement in the brain, and that low birth weight infants have a very small body fat content. And this fat is what is in part being used to help supply the ketones for the brain. And that means that those babies with a low birth weight have a higher risk of neurodevelopmental delay. And this, of course, has an impact, a lifelong impact. If there are problems with development of the brain during infancy, the impact is lifelong. So this has an importance for trying to understand how to protect the brain as we're getting older. So I come to this model of the hybrid car because this is especially true in the infant brain. So the brain is using, the car is using gasoline and it's using electricity, and it's alternating between both constantly. The car's computer is deciding which one it will be using. There's no computer in our brain, but the glucose is an important fuel, and ketones as well, and there's a cycling process, which is especially important during infancy. So just to remind you about ketones or ketone bodies, they're derived from dietary fats, from stored fatty acids, and nowadays they can also be obtained from salts or esters. The main ketone, the parent ketone produced in the liver is acetoacetate with a ketone bond here. And it is in equilibrium with beta-hydroxybutyrate. So the ketone becomes a hydroxy group in beta-hydroxybutyrate. Some acetoacetate is decarboxylated to acetone, which is in the blood and will also appear on the breath in people in relatively high ketosis. So if we come back to ketones and the infant brain, and I reported the overall story here in relation to infants in the Journal of Human Evolution, because we feel it has an impact on the development of our species over the past 2 million years. In infants, ketones are essential to supply 25 to 30% of the brain's energy needs constantly. This is in a normal, well-fed infant. This is not an infant that is sick, malnourished, low birth weight, or any such issues. This is a normal requirement for the brain 24 hours a day, 7 days a week. Ketones are also supplying 90% of the carbon used to make brain lipids, especially cholesterol and myelin lipids. In this image, the white part is literally the myelin, and it is insulating the neurons and the axons. And that insulation is made of cholesterol and other lipids that are made in the brain, and they're made primarily from ketones. So the infant brain is using ketones to both to build the structure of the brain and also to fuel the energy requirements. So to show you this in a little bit more detail, in the image of the brain, we can show you what one of the tracks looks like. So these are connections between the cortex of the brain and the temporal lobe here, for instance. And these lipids are not coming from outside the brain. The cholesterol and saturated and monounsaturated fatty acids, there's almost no uptake from outside the brain, so they bounce off the brain. They're coming from ketones as the primary substrate. Glucose is also a substrate, but ketones are the primary substrate, and they're derived particularly from medium-chain triglycerides, MCT, also from some beta oxidation of polyunsaturated fatty acids, perhaps surprisingly, linoleic acid, alpha-linolenic acid, which can be used to make omega-3 fatty acids used by the brain, but they're also used to generate ketones, which will then be used to make the myelin lipids. So ketones are an essential part of early brain development. They are produced, as I mentioned earlier, from medium-chain triglycerides in milk of all mammals. And their absorption is more rapid than the normal absorption of lipids in the diet. So the route for common dietary fatty acids is the number one, which goes through the lymphatic system, the peripheral circulation, chylomicrons, and eventually gets to the liver and many other tissues. The ketones are produced from medium-chain triglycerides, which are absorbed directly up the portal vein, directly to the liver, making a rapid source of ketones, which are helping supply the brain. So this is a very special situation where these nutrients are coming through a special rapid route to the liver compared to the normal dietary fatty acids, long-chain fatty acids. And the final point is that some of those medium-chain triglycerides in milk are going into body fat in the infant. So they extend the ketone exposure of the infant beyond the end of lactation. Once lactation stops, the MCT in milk will no longer be present, but there will be some in fat. And these are magnetic resonance images of transections of an infant. So they're just images. There's not much fat, of course, around the skull, but the cheeks have fat layers. The chest has a lot of fat. The arms, the buttocks, and the thighs of the infant. And so there's a lot of subcutaneous fat which is available to supply the fatty acids necessary to make importance for ketones, a need for ketones, and it is well equipped to obtain and use them. The second important point that I want to mention is that the two main fuels of the brain are taken up by two different strategies. So if you imagine the blood supply here on the left and the brain here on the right, it is the utilization of glucose in an area of the brain that to make ATP. And when that glucose concentration goes down in brain cells, they are stimulated to increase glucose transport and to pull the glucose into the brain from the blood. So you can have a high amount of glucose in the blood after eating some dessert or some refined carbohydrate. It will not increase the amount of glucose going into the brain. Ketones is the opposite story. When ketones are elevated in the blood, it's usually a signal that insulin is low, that glucose is low, and that the brain will need ketones rapidly. And they are pushed into the brain in relation to the concentration in the blood through the monocarboxylic acid transporter. Unfortunately, this is the same abbreviation as medium-chain triglyceride, but I won't be using it again to talk about the transporter. The third important point is that ketones entering the brain are able to spare brain glucose. So we can assess this relationship by imaging with ketone PET imaging and ketone glucose imaging, as shown in this small image here on the right. And when you make these measurements before a ketogenic diet and after a ketogenic diet, you can see that the brain uptake of ketones goes up. The delta ketones is going to +3, +4, +5, +6 above the baseline values. And as the ketone levels go up, the uptake of glucose goes down. And this depends on the area of the brain that's affected. So the subcortical nuclei very deep in the brain have a less ketone uptake and less decrease in glucose. But the frontal cortex, for instance, here and here, has a higher ketone uptake and a more significant decrease in glucose. And this is a strong correlation, inverse correlation. So ketones can help conserve glucose for other purposes. This relationship between plasma levels and brain uptake is shown on the graph here, where plasma ketones are shown on the x-axis, and two different ways of looking at the brain uptake are shown on the y-axis here to the left and the y-axis here to the right. So here on the right, we're showing ketones as a percentage of brain energy metabolism, and here we're showing the actual units of ketone uptake. So in the postprandial state after a meal, ketones are low, and ketones are contributing a small percent to brain energy metabolism. If you fast for 24 or 48 hours, ketone levels in the plasma will be increased, probably 10 to maybe 100-fold, and they will be contributing about 10% to brain energy metabolism. And then in some extraordinary studies in the past, which included fasts of 40 and even 60 days, you can see that the contribution is above 70% to brain energy metabolism. So it's directly proportional to plasma concentration over a high range. Again, I come back to this point because with the images, because we use ketone PET imaging to demonstrate that in the early, before we start the ketogenic diet, ketones are contributing about 3% of brain energy. So this blue area, very low, represents about 3% of brain energy metabolism from ketones. And then on the ketogenic diet for just 4 days, the ketones are contributing about 30% of brain energy metabolism. So everything turns to orange and red. So this is a simple way to measure ketone metabolism, and we quantify it carefully with the necessary blood samples. So the topic that we want to address in relation to cognition and aging is Alzheimer's disease. And you can see here that there's a problem in Alzheimer's disease because the color is going down from the bright red and orange that you should see above the ears in the control person, the person with Alzheimer's disease. There is no orange or red. So this means that the glucose uptake in the brain is significantly decreased. This problem has been known for nearly 40 years now. Traditionally, this glucose deficit in the Alzheimer's brain has been considered to be a consequence of the disease. So we see the deficit in the parietal cortex, where there's no red or orange, compared to the frontal cortex, where we see still normal uptake of glucose. So when we say consequence, we mean that traditionally people have felt that the neuropathology, the beta amyloid, the senile plaques in the brain are causing cell death, is causing the cognitive decline. And because there are fewer cells and less communication in the brain, this is reducing the brain glucose uptake. We view it differently. We view this problem as a contribution to Alzheimer's disease, not a consequence, but a contribution. So instead of having the starting point in this problem being with the neuropathology, we're suggesting that the problem starts with low brain glucose status, specifically before cognitive decline starts. This then helps develop the neuropathology, which leads to the cognitive decline, which further lowers brain glucose uptake. And you end up with a vicious cycle leading to brain energy exhaustion and the full-blown clinical syndrome of Alzheimer's disease. So what is the basis? What is the evidence for low brain glucose status before cognitive decline? Here I show you 1, 2, 3, 4, 5, 6 examples of people with low brain glucose status before cognitive decline starts. This is in healthy older people, in people with a family history of Alzheimer's disease, in insulin resistance as young as 25 years old, in ApoE4 carriers as young as 30 years old, even in the presenilin mutation carriers who are predestined to get Alzheimer's disease, and in traumatic brain injury. In all these cases, even in the presenilin carriers, the low brain glucose energy status has been demonstrated before the onset of cognitive decline. So it cannot just be a consequence of the disease. It is probably contributing to this process. So we talked about the hybrid car, and all I've talked to you about in this sequence of slides about this concept is glucose. So clearly, it would be useful to know what is happening with ketones as well. Brain ketone uptake, potential for rescue, and for improving brain function. So in the review that we published this year, Nature Reviews Drug Discovery, I summarize this hypothesis, and this particular image is shown in that report as well. So what we're showing here is glucose uptake, the uptake rate constant for glucose in Alzheimer's disease in controls, healthy age match controls, mild cognitive impairment, and Alzheimer's disease. So you have a wide distribution of the orange to yellow color in the healthy controls, and in the mild cognitive impairment, the orange starts to go down in the area above the ears, and this is severely reduced in Alzheimer's disease, now down in the green. No orange to be seen anywhere in the brain. So the upper level where you should normally be seeing lots of orange to reddish color is gone. This is a different form of the same PET image I showed you earlier. It's been known for a long time. What we've done is to look at the same people, exactly the same people. 2 hours before they had this scan, they had a ketone scan as well. And when we look at the uptake rate constant for ketones, it's lower. It starts out lower because ketones are normally contributing 3 to 5% to brain energy metabolism, while glucose is contributing 90 to 95% or even more. But in MCI, as you change to MCI, you can see there's no ketone uptake. It's even a little more yellow here, and in the could get to Alzheimer's disease, there's even some orange showing, which is suggesting that the actual transport capacity to get ketones into the brain is not decreased at all in the same individuals. It's actually increasing slightly, or well, no change. Statistically, there's no change, but in some individuals, we do see a small increase. So if these cells were dead, they would not be able to take up the ketones. This is an active transport process. The fact that they can take up ketones but not so much glucose suggests that there is a specific problem with glucose. And when we plot these results in relation to plasma ketone levels and brain ketone uptake against the normal curve for healthy individuals in a postprandial state or fasting, or even the long-term fasting, as I showed you earlier, people with Alzheimer's disease and MCI are on exactly the same slope. The Alzheimer's disease ones are in black, the MCI in red, and our tracer is acetoacetate, so it's slightly off not exactly the same line for beta hydroxybutyrate, which is normal. So the capacity to take up ketones in mild cognitive impairment, the brain's capacity is the same in Alzheimer's MCI as in controls. So if we come back to our hybrid car, the rest of the talk will be about the BENEFIC trial, which is on clinicaltrials.gov. Part of it was published in Alzheimer's Dementia last year, 2019, and part of it has just been accepted for publication in Alzheimer's and Dementia, and will be online before the end of October. The goal of this study was to see whether improving energy, brain energy rescue, was going to improve cognitive outcomes in MCI. Remember, glucose should normally be supplying 70 to 95% of the brain energy requirements, and ketones 5 to 30%. But in MCI, the decrease is about 10%, and early in Alzheimer's, it's about 20%. So this is a constant energy deficit in Alzheimer's disease every day of their lives. Here, there's no decrease, and it appears that this side can work as an excellent fuel. So the design of this trial is that, the design of this trial is on clinicaltrials.gov, and the study was separated into two phases. The first part was funded by the Alzheimer's Association and involved the brain imaging. The second part was not funded by the Alzheimer's Association and did not involve brain imaging, but a metabolic study. What was constant between the two phases was the cognitive evaluation before the intervention and the same cognitive evaluation at the end of the intervention. And the study was done over 6 months with a daily oral nutritional supplement taken at breakfast and at supper. The nutritional supplement was based on a skim milk emulsion of medium-chain triglyceride, C8 and C10, in whom 39 people completed the trial, or placebo, in which high oleic acid sunflower oil was the active ingredient, in which 43 people completed the trial. So you can see here that the data are based on 82 people. We had 122 people that were recruited. So we had a completion rate of about 75%. My slides are not advancing. I apologize. Can you hear me? My slides are not advancing.
Yes, I can hear you.
Thank you. Unfortunately, something is, okay. So the first results of the BENEFIC trial I would like to show you is that when people took the ketogenic medium chain triglyceride, there was a sustained blood ketone response. So this means that it's a bit small, but here at the bottom is the placebo, and obviously there was no ketone response to the placebo when it was given at time 0, either before or the white squares are for before the trial started, and these are at the end, and they might look different, but they're not statistically different. So the ketone response to the MCT is identical over 6 months. There's no loss of the ability to make ketones from MCT over 6 months. And what this shows on the right is that there's a twofold increase in brain ketone levels shown by the PET ketone imaging. So a low ketone uptake before they start the trial, and on the last day of the trial, using the PET imaging, we can show the ketones lighting up in the brain if they were on the ketogenic MCT, but not on the placebo. The cognitive results are summarized here. The outcomes improved on 4 cognitive domain areas. This will be the citation when it comes out at the end of October. So of the 4 cognitive domains, episodic memory is shown here, executive function is shown, and language. And I'm not showing the processing speed. It's going to be in the paper. So for episodic memory, it's the free and cued recall test, especially trial 1. And we're showing the delta words, which means the change in numbers of words recalled between the start, the baseline of the study, and the end of the study. So on the placebo in green, 43 people are shown here, and the mean, it changes 0. Whereas there's a 1-word improvement in the KMCT group. So of 16 words they have to recall, they've been able to recall 1 more on average than the placebo. For the executive function, it is verbal fluency, where there's a decrease in the placebo group, a slight decrease of 1 word, and a 2-word improvement in the ketogenic MCT group. So a difference between them of 3 words, highly statistically significant. And on the Boston naming test for language, the total number of correct responses did not change on placebo, but improved on the ketogenic MCT by 1.3 words, also statistically significant. We looked at the relationship between the cognitive outcomes and the plasma ketones. Remember, this study was done in 2 phases. We had PET imaging for the first phase, so only half the participants have PET images, but we have plasma ketones for everybody, almost everybody. So we looked at the change in cognitive outcomes against the change in plasma ketones for the same test: episodic memory, executive function, and language. Again, the change in words on the y-axis for each of the 3 tests, and the change in beta hydroxybutyrate on the x-axis. And 0 would mean there was no increase in ketones. Intervention had an improvement in ketones after the test, whereas the people on the placebo are all around 0 or below. Same thing for the executive function and language. And there's a statistically significant positive relationship between the change in number of words recalled and the change in plasma ketones, or for verbal fluency, or again for number of words for the language test. In each case, the p-values on the order of 0.3, 0.1, 0.4. So we're seeing that as you increase the ketones, you have a better chance of increasing the word recall necessary. This also suggests that ketogenic MCT is not that effective in raising ketones in everybody. There's a lot of people that are still here at low values. So a higher dose of KMCT would probably help extend the curve out a little further, or a different form of ketone supplement. I want to add these results, which are about to be submitted for publication, which is the cognitive outcomes in relation to ketone uptake in the white matter tracks. So what you're seeing here in the bar graph is the coefficient of correlation between the ketone uptake in these tracks and processing speed, the rapidity with which you can respond to the test that you have to complete. And the colors here correspond to the colors of the tracks in the brain. I won't bother with you with the names of these tracks, but the ones with the stars, so the blue one, the red one, the green one, the pink one, and this blue one as well. In most of these regions, but not all of them, in most of these tracks, there is increased ketone uptake and a very strong correlation with processing speed. Here's a correlation of 0.5, and some of them are up over 0.6. Remember, these are differences, difference in ketone uptake and difference in performance on this cognitive test. So this is a remarkably good outcome, and it suggests that the quality of the white matter helps improve the quality of memory and the ability of cognition in mild cognitive impairment. Now, we have talked about the problem with glucose, and I refer to it as the brain energy gap in the Nature Reviews review that we published this summer. So this gap here refers to the difference between 100%, which is in the healthy young controls, and those with MCI. So the gap here, you can see it's just below 90, it's about 88%. So there's a 12% gap, which is shown here at the bottom. The gap is shown at the bottom. So young people have no gap, and their brains are running about 5% on ketones, 95% on glucose. The healthy older controls, they're still cognitively normal, but they actually have a brain energy gap of about 8%. It's partly due to lower glucose uptake, and it's partly due to lower ketone uptake. This gap gets bigger as you get mild cognitive impairment, where the glucose uptake goes down to 85% from 89%, but the ketone uptake remains at 3%. So the gap is 12% of where it should be. And from the study, from Benefic study with 30 grams a day of MCT, you can see that we have a bigger bar here in orange. The ketone uptake is contributing to reduce the brain energy gap from 12% to 9%, because the ketone contribution has gone from 3% to 7%. It's more than double. And we would predict, so this is in brackets, but we would predict that the uptake of ketones would increase further if you were to give 45 grams a day of medium chain triglyceride. There again, the glucose problem would not change, but we would reduce the gap because we would increase ketone contribution even further. So I've discussed our experience in one study so far. But over the past 10 years, you can see Henderson published in 2009, there have been several reports documented on the clinical trials website as well, both in Alzheimer's disease and in mild cognitive impairment and in subjective memory complaints. Some have been randomized controlled trials with MCT, some have been randomized controlled trials with ketogenic diet, some have been open label studies, and some have been crossover studies. Duration here, 6 weeks, others like ours, 6 months, 12 weeks, 4 weeks, quite variable. Some had cognition as an endpoint, some had just feasibility as an endpoint. Is it feasible to give a ketogenic diet to people with MCI? You have to do the study to find out. Does it change brain glucose and ketone status? So cognition was not an endpoint, it was brain energy metabolism. So this is to say that our study that I've just reported to you is essentially consistent with and perhaps a little better results than some of these studies, because we had enough people to evaluate cognition. We had enough time, 6 months, to assess cognition. And we had a study done in MCI that was randomized and controlled against placebo. So it was robust enough to objectively assess cognition. So I would like to summarize my presentation by showing you the same image, which is an old image now from it's been 40 years that we've known that there's a problem with glucose uptake in Alzheimer's disease. It's not a consequence of the disease. It's a pre-symptomatic problem that is specific to glucose and is causing brain energy deficit. It is possible to provide more energy to the brain by ketones. This is feasible both in MCI and we've published in both areas. This approach with brain energy rescue recapitulates the physiological role of ketones in the infant brain. So it is simply, we are trying to reproduce in older people what nature and our evolution has shown us was necessary for a long, long time. It's clear that we're making some progress, but the cognitive outcomes still need to be further evaluated in larger randomized controlled trials. More work is needed on the mechanism of action. Is it simply as a fuel? Is it because we're improving myelination in the brain? Are we reducing neuroinflammation? I'll just go back for a moment to this slide where they showed that the cerebrospinal fluid A beta 42 levels were increased on a ketogenic diet in MCI. This is a sign that the brain is able to get rid of some of the neuropathological material in the brain. So what is the mechanism? Are we actually reducing the neuropathology? What is the optimal ketogenic strategy to use. And I haven't said anything about other neurodegenerative diseases of aging, but there is some indication that ketones can help Parkinson's disease as well. So what we've discussed here is really a prevention approach in which we use ketones to help support brain energy metabolism, but exercise would surely be beneficial as well. Probably other lifestyle improvements that could help, perhaps a Mediterranean diet, obviously stopping smoking, social life is very important as well. And so as to reduce the risk factors for Alzheimer's disease, including diabetes, hypertension, and obesity. So thank you for joining me in this webinar this evening. And thank you to the research team that did all this wonderful work, to my collaborators, the geriatricians, neurologists, neuropsychologists, nurses. Everyone has been played a key role in generating these results. The participants have been so willing and so active to have blood samples and imaging and cognitive tests and carry home their monthly supply of ketogenic MCT bottles. So they've been a terrific help to us and to our partners for the funding and in-kind materials used in this study. Thank you again.
Well, thank you very much, Dr. Cunnane, for this outstanding presentation. Very interesting. We have some questions here. The MCI participants in your RCT, the Benefic one, had MCI due to Alzheimer's disease? If yes, do you have data on biomarker profile?
Yes, I did not give the demographics, but they're in the paper. And so MCI, people with amnestic cognitive problems, memory problems, are more likely to progress to Alzheimer's disease. 50% of our MCI population had episodic memory problems. The other 50% did not, or had other problems as well. So not everyone is necessarily going to get Alzheimer's disease from this population. We did not have A-beta levels in the brain or phospho-tau. It's an even more expensive study to do. It's more complicated for this population. So we relied strictly on the cognitive outcomes or the cognitive performance at the beginning to choose our patients.
Would insulin resistance interfere in the production of ketone bodies when MCT is consumed?
Will insulin resistance interfere?
Yes. Yes.
That's an excellent question, and the answer is probably not. Insulin resistance does not seem to. We have done one study which we published last year in pre-diabetics, showing that they are still able to generate approximately the same amount of ketones in the blood from the same dose of MCT as someone who is not diabetic. If you have maybe more advanced diabetes, there may be a problem, but MCT can seem to go around the insulin-dependent steps in ketone production. So I think we should be able to use MCT in people who have mild insulin resistance, but it has not been adequately studied yet, especially we don't know if we can improve cognition, for instance, with MCT in people who have insulin resistance. The people in our study did not have insulin resistance. They had normal blood glucose.
Does MCT provide ketone bodies without fasting or carbohydrate privation?
Yes, definitely. This is the example I showed is that MCT will produce it even if you have a bad diet, actually. You know, lots of refined carbohydrate. MCT will produce ketones. So we did not try to change the diet of the participants in the study, it is still an effective way to produce ketones.
Actually, another question that you have already answered in your presentation, but when you say MCT, which fat acid do you refer to?
Yes, it's complicated because we can go from 6, 8, 10, 12, 14. We're talking about 8 and 10 carbons. 8 carbon MCT are more ketogenic than 10 carbon, and 10 carbon is more than C12 and C14. So the important ones for ketone production are C8 and C10. The problem is that most products, we don't necessarily know the content of C8 and C10, so it can be difficult for the members of the public to make the right choice.
Did you employ any global cognitive measure?
I didn't hear your question very well.
Did you employ any global cognitive measure?
Yes, indeed. We use the MMSE and the MOCA, the MOCA, the Montreal Cognitive Assessment, for screening purposes. We did not use it as an evaluation of cognition at the end.
Regarding amount of fat from the active and the placebo group, why were they different? 15 grams versus 13 grams. How this impact the results?
I don't think it impacted the results because 13 grams of a long chain fatty acid at 9 kilocalories per gram is the same energy value as 15 grams of MCT, which has an energy value of about 7 kilocalories per gram. So we're giving the same energy value of the two types of fat.
Did you see any difference between patients with familiar and sporadic Alzheimer's disease?
Well, they didn't have Alzheimer's disease. They had mild cognitive impairment. So we can't answer that question.
Regarding the source of MCT, you already answered C8 and C10 only.
The supplier it's not publicly available. So the supplier, we bought this wholesale. It was a gift from Abitec Corporation. But I'm sure you could suggest somewhere how to get it in Brazil, but that was our supplier.
Okay, but the composition was only C8 and C10.
Yes, 55% C8 and 35% C10 and some residual other fatty acids.
Okay, an interesting question. It's about the cell type or types responsible for the ketone uptake in the brain. Are they neurons or glial cells, or which one?
We don't know. The simple answer is we don't know. The neurons have to metabolize the ketones. Whether they're getting them from the astrocytes or whether they're getting directly, we can't tell you from PET imaging. It's impossible to know. The oligodendrocytes are probably metabolizing ketones as well because, as I mentioned, there's quite an increased performance of the processing speed, which is dependent on the oligodendrocytes and the myelination. But the short answer is I can't answer from PET imagings. We don't know.
Dr. Ferreira is contributing, saying that in the case of glucose uptake, recent studies suggest that the most uptakes by astrocytes.
Yes, and I don't challenge that. It could be that the astrocytes are taking up the ketones predominantly and transporting them to the neurons. It hasn't been studied.
Oh, is it the case? Maybe it's a common question. Is it the case to consume MCT as prevention?
Is it the case? No.
No. If maybe you recommend the consumption of MCT as prevention.
I'm not. I don't wish to make a recommendation because I don't want to oversimplify the nature of the problem. I think they are part of the solution, but they will be. It would be important to try to do regular exercise. It would be important to treat hypertension. It would be important to not have insulin resistance, hence the reason for the exercise. It's very important to have a good social environment as much as possible. And some of the anxiety and stress that we might have in life, MCT will not change that. Ketones will not change that. So this is a disease that develops over 20, 30, 40 years. MCT can be part of the solution, but I would not want to leave the impression that that's all you need in order to prevent Alzheimer's disease. I think that is naive.
And could you comment about the safety of MCT?
Yes, again, I didn't have time to discuss it, but we looked at the safety profile in terms of metabolic. So triglycerides do not go up, body weight does not go up, body fat content, waist circumference does not change, cholesterol doesn't change, insulin, glycosylated hemoglobin doesn't change. So in general, the cardiometabolic profile is very good. And I know that people are concerned about saturated fat for cardiovascular health, but there's no publication in the literature that I'm aware of that suggests that MCT are a cardiometabolic risk. And our data are from people of 72 years old, taking it for 6 months at 30 grams a day. I think that's a fairly good level of evidence that there is no cardiometabolic risk associated with MCT.
Do you have the ApoE4 data from the participants?
Yes, they were evenly split between the placebo group and the active group. About 25% of both groups had ApoE4, at least one allele, and we can see no difference in any of the major outcomes between whether they were a carrier of ApoE4 or not. But the project was not powered to assess that adequately. So it's hard to know. There may be some effect of ApoE4 on some parameters, but it's still hard to say.
You mentioned in your presentation, but which is the difference between glucose metabolism in a normal aging brain versus the Alzheimer's one?
Yeah, so in normal aging, in fact, the glucose problem is on the front of the brain, going down about 7 or 8% in the frontal cortex, whereas in the Alzheimer brain, the problem is on what they call the parietal and temporal lobes, the side of the brain. So does it mean that those people with a problem here, gradually the problem will spread to the parietal lobe? We haven't done a longitudinal study, so we can't say. But it is it's both the location of the problem and the magnitude of the problem. Because in healthy older people, we see about 8% deficit, 7-8%. Whereas in early Alzheimer's, it's about 20%. So much higher, and it gets worse and worse as Alzheimer's disease continues.
Safety, you already answered. Tolerability? Could you comment on?
As I mentioned during the methodology part of the talk, we had about 78% of the people who started the study were able to complete it. So that's not too bad for this type of product. It was one that we had made locally, but it was not, we are not food scientists or emulsion specialists, so we did the best we could, but I think a better emulsion process would have had more people complete the study, I think, because MCT is still associated with stomach discomfort, and so we start with a smaller dose over the first 2 weeks, 10 grams a day, 15 grams, 20, up to 30 grams a day. We don't start at 30. We start at 10 and go up to 30.
Okay, so I think that's it. We don't have more questions. So, Dr. Cunnane, thank you very much again for being here with us. It was a huge pleasure.
The pleasure was mine also. I hope it's helpful. And if you get some more questions, I'd be happy to try to answer them. Helen, thank you for the invitation.
Perfect. Thank you.
And good luck and bye-bye.
Thank you. Bye-bye.
Thank you. Bye-bye.
And I'd like also to thank you all to join me with us this international webinar this evening, and see you in another opportunity. Thank you very much.